first case, I will showcase how we utilize DMARS second nanodisc and peptide nanodisc technology to screen loop-specific monoclonal antibody, especially for, in this case, is ECL1, extracellular loop 1. So Claudin 18.2 is an gene is like a specifically overexpressed or like exposed, this protein exposed in the gastric or pancreatic tumors. And in the normal case, they also express in the junction part. They are not exposed for the antibody. So when in the tumor case, they will overexpress and expose. And also there's an isoform for Claudin 18.2 is 18.1. So these two genes come from the same, these two proteins come from the same gene through alternative splicing. The only difference on the right side, you can see the only difference from 18.2 and 18.1 is their extracellular loop 1 on the extracellular side. So how to really develop an antibody only recognize ECL1, but not ECL1 for the Claudin 18.2, not 18.1 is a pretty challenge because you have to develop an antibody only recognize 18.2. So you only have one choice of develop an antibody against ECL1. And the difference only 9 amino acid. So in this case, we start generate functional proteins and a cell line for the immunization and the screening. So we develop a K562 cell line overexpress Claudin 18.2, and also we made second nanodisc, peptide nanodisc for the 18.2, and also we made 18.1 peptide nanodisc for the downstream screening. And so during the initial immunization schedule, you can see initially we use a stable cell line, so they didn't generate a good enough serum titer. Then we start to boost them with nanodisc, purified nanodisc like 18.2. After immediately after boost with nanodisc, the serum titer get boost up. So over the course, we constantly we did several rounds of nanodisc immunization. And so this is the screening approach. We utilized the peptide nanodisc for 18.2 to do the depletion. So any cross-react with the Claudin 18.1, we use peptide nanodisc 18.1 to get rid of them. The remaining B cells, then we go through another column of the Bs with a Claudin 18.2 peptide nanodisc. So only those specifically recognized Claudin 18.2 B cell will be enriched. Those cross-react with 18.1 will be get rid of in the first round of peptide nanodisc for 18.1. So after we disseminate in this 18.2 specific B cell into 90 plate and clone them, and we successfully got a couple of clones. Here is the functional evaluation. So all these 18.2 specific clones, we made them as a CAR T cell, and so we test with K562 Claudin 18.2 target cell, and also the control we use Claudin 18.1 transfected stable cell line K562. So from this CAR T tumor CAR T cell killing test, you can see our 18.2 specific mAb converted CAR T cell shows a very specific to cell killing effects. And BMK, we like a benchmark, we use CT041. This is a benchmark in clinical trial. This is 4 of our clones showing comparable functionality and comparing to the benchmark antibody and also specificity.